My name is Vriti Mirchandani, and my project is called Novel Photochemically Activated Quantum Dot Conjugates for Modulation of Neurodegenerative Protein Aggregates. Neurodegenerative diseases like Alzheimer's and Parkinson's affect millions of people worldwide, and despite decades of research, one major challenge still remains: protein aggregation. Proteins normally fold into stable structures that allow them to function properly, but under certain conditions, proteins can misfold. Those altered proteins begin interacting with each other and gradually assemble into larger and larger structures until stable aggregates form. And once those aggregates form, they become extremely difficult to remove. They resist normal cellular clearance mechanisms and continue interfering with surrounding functions. What makes this especially challenging is that toxicity depends heavily on structure. Not every protein is harmful, but certain aggregated conformations become problematic. Current approaches try to address this problem in different ways. Some aim to slow aggregation, some attempt actual targeted removal, some use nanoparticles to actually interact with the proteins, but many still face the same limitations: limited precision, limited time control, and limited selectivity. And that made me start thinking differently. Instead of asking, how do we constantly remove aggregates, I started asking, what if disruption itself became controllable? Could we create a system that stays inactive until we intentionally inactivate it? That became the central idea behind this product. This slide shows the mechanism I designed to test that idea. The starting point is already formed protein aggregates right here. Then I introduced them to quantum dots. Quantum dots are semiconductor nanoparticles commonly known for imaging and fluorescence applications, but what interested me was another property: their ability to become chemically active after photoexcitation. The idea wasn't continuous treatment. Instead, first allow the quantum dots to associate with the aggregates, then apply blue light. Once activated, the quantum dots generate localized reactive oxygen species, or ROS. Those reactive species were expected to destabilize the aggregate structure. To determine whether disruption occurred, I quantified the change using fluorescence. So conceptually, the system behaves almost like a switch. Without light, nothing happens, but with light, activation occurs. So, my hypothesis was, if quantum dots associated with aggregated proteins, then optical activation would induce localized chemical activity that selectively destabilized aggregated conformations while minimizing effects on native proteins. So the next question became, how does this actually happen experimentally? To test my hypothesis, I developed a controlled in vitro experimental model designed to isolate the effect of photoactivated quantum dots on protein aggregation. First, I generated reproducible protein aggregates using thermally aggregated egg albumin. Egg albumin was selected because it provides a constant aggregation behavior while allowing aggregation dynamics to be studied before moving into actually disease-relevant proteins like tau or alpha-synuclein. Next, I introduced CDSE or ZNS quantum dots and allowed them time to interact with the aggregated protein. After incubation, samples were divided into illuminated and dark conditions. For activation, I exposed samples to 470 nanometer blue light. To quantify changes in aggregation states, I used cypro-orange fluorescence dye. Cypro-orange binds to the hydrophobic regions that become exposed during aggregation, meaning higher fluorescence corresponds to greater aggregation, and lower fluorescence suggests aggregate destabilization. To strengthen interpretation, all experiments were performed under 3 independent biological replicates, while technical triplicates. At this point, the system was built. The next question became how to structure the experiment to determine whether disruption actually occurred and whether it was activation dependent. This slide shows how I organized and interpreted the experimental conditions. On the left is a plate layout used to separate all treatment groups. I included aggregate controls, quantum dot controls, native protein controls, dark conditions, and illuminated conditions. This setup allowed for direct comparison across conditions while isolating the effect of optical activation. On the right is the proposed mechanism behind disruption. In the absence of light, aggregates remain largely intact, even when the quantum dots are present near the surface of the protein. However, under 470 nanometer illumination, the quantum dots become activated and generate localized reactive oxygen species, or ROS, and that ROS is expected to destabilize the aggregate structure, producing smaller and less ordered assemblies, as shown. Importantly, the goal wasn't to complete protein destruction, but was to control the modulation of the aggregation state. The design allowed me to test whether observed changes were truly caused by photoactivation or rather than just concentration effects. Background fluorescence or passive nanoparticle interaction was also included. With the system and controls established, I could finally test whether activation actually changed aggregation. And so that brings us to the results. This became the central experiment of the project. If the hypothesis was correct, disruption should only occur when both quantum dots and light were present. And that's exactly what I observed. At the native state of the protein, there was very little fluorescence, indicating very little aggregation, which is consistent. Then, with the aggregated protein, which was the heat-treated egg albumin, we see a rise in fluorescence, indicating a rise in aggregation. This is still consistent with the results we already know. Then, when using quantum dots but not treating it with light at the moment, we see a very little decline in fluorescence, indicating a small decline in aggregation. So ROS is still not being generated to its full capacity. But when we finally expose the quantum dots to light, therefore turning on the switch, we see a huge decrease in fluorescence, indicating a 44% decrease in aggregate fluorescence, indicating that ROS has been generated. So, the reduction only occurred when both conditions were combined, and this suggested that disruption was activation dependent rather than continuously active. At this point, I knew something meaningful was happening, but I still needed to understand why, so I designed additional experiments. After observing the initial reduction, I ran additional experiments to determine whether the effect was reproducible and to better understand the mechanism behind it. First, I tested concentration. I found that disruption increased as quantum dot concentrations increased, but eventually plateaued. So this suggested a saturable response rather than random signaling reduction. Next, I investigated timing. Most disruption occurred rapidly within the first 10 minutes, but eventually stabilized. So that suggested that efficient activation rather than gradual degradation. Then I wanted to understand the mechanism. So to test that, I measured the reactive oxygen species, and photoactive samples produced substantially greater ROS signals compared to dark controls. That supported the photochemical explanation, and at the same time, I monitored temperature. Temperature remained stable throughout the activation, suggesting that disruption wasn't driven by heating. I also tested different wavelengths, and I saw that maximum disruption occurred specifically at 470 nanometers, which aligned with the expected quantum dot excitation behavior. Finally, I tested selectivity. When native proteins underwent the same treatment, fluorescence remained essentially unchanged. Meanwhile, aggregated proteins demonstrated strong disruption. Together, these additional experiments helped establish that the observed effect was not only reproducible, but appeared to be rapid, activation dependent, ROS associated, and selective for aggregation conformations. So overall, this project demonstrates that photochemically activated quantum dots can selectively modulate protein aggregation in a light-dependent manner. Rather than continuously acting on proteins, disruption only occurred following external optical activation. Importantly, aggregate disruption occurred while preserving native proteins under the tested conditions. At the same time, there are important limitations to note. The study was performed using an in vitro aggregation model, so we used egg albumin instead of the actual disease-relevant proteins, such as tau or alpha-synuclein. This was largely due to the fact that we were able to replicate the beta structures of the disease-relevant proteins in 15 minutes instead of weeks, which was valuable time we didn't have in the lab. Additionally, structural changes were inferred through fluorescence rather than directly visualized. So in the future, steps could be taken such as conducting an SDS page, PEM, or DLS in order to actually visualize the disaggregation in the protein. Additionally, optical activation conditions weren't fully reflecting the tissue-level light penetration. So I don't view this work as a therapeutic solution. I view it as establishing a framework that explores whether protein aggregation, traditionally viewed as a static pathological endpoint, can actually instead become a dynamic, controllable biological process. So thank you for listening. See you next time.